Kia ora koutou, ko Sue Brewster, tako ingoa. Well, good evening everybody. My name is Sue Brewster and I am the Executive Director of Auckland Medical Research Foundation. So we have been around since 1955 and we are the largest independent funder of medical research in New Zealand, and that is across the spectrum of all diseases, disorders and conditions. So the thing that donors love about us is that all of our administration expenses are covered by a very generous benefactor, and so that means that every cent of every donation goes directly into our purpose, and that is funding medical research. And since 1955 we have funded over $92 million worth of medical research, and that's thanks to you, all of our generous donors out there who have contributed and helped to make such a significant difference in so many people's lives. So tonight I want to welcome you to one of our online events, and tonight's focus is the latest in cancer research. So we're very fortunate to be joined by Professor Peter Browett and Associate Professor Nuala Helsby. Tonight, we're going to hear from Peter about his research into the latest cancer therapies, and particularly leukemia, which is a specialty area, and how they're trying to look at individualising treatments. And from Nuala, we're going to hear about how environmental and inherited factors influence cancer treatments. Professor Peter Browett wears many hats, and he has been on our medical committee for many years and is also the chair, and he's also been on our AMRF board. He is the Professor of Medical and Clinical at the University of Auckland in the Molecular Medicine and Pathology Department. He's also the Director of the University Research Centre for Cancer Research. And he's also a clinician at Auckland Hospital. So we're very fortunate to be hearing from Peter tonight because, as you can imagine, he is a very busy man. Not unlike Nuala, Associate Professor in the Pathology Department at the University of Auckland, and also Honorary Associate Professor of the Auckland Cancer Society Research Centre. And also on our medical committee. So we're very fortunate to have such talented experts on our medical committee and helping to make those funding decisions. So tonight you're going to hear from both of them, and we'll do the presentations back to back. So we'll hear from Peter first, followed by Nuala. And please, there will be time for questions at the end. We'll have them live, so you can ask any questions you like. You can pop it into the chat box that you'll see on screen, and then we will cross to them live after the presentations. The presentations are about 30 minutes each, so please, you can sit back and relax, and there will be some technical parts to these presentations, so please save up those questions for the end. Sit back, relax, and here's Professor Peter Browett for his latest in cancer research. Thank you. Kia ora.
My name is Peter Browett. I'm Director for the Centre for Cancer Research at the University of Auckland. I'm also a clinical haematologist at Auckland Hospital, and my research interests are around the genetics of leukemia. And today I have an opportunity to talk to you about our research on personalised haematology, and this is really our goal is to improve outcomes for patients with leukemia, with giving the right treatment to the right patient at the right time. If we look at the whole area of cancer, one of the biggest advances over the last decade has been the understanding of the genetics of cancer and looking at the changes that occur in the genome of cancer cells. And this has allowed us to help with the classification of cancers, but also looking at potential targets for therapies. And so the goal of our research is to assess the impact of genomic testing and the management of patients with blood cancer. So this is the clinical problem that we're dealing with. So I look after patients with acute leukemia. So on the left-hand panel, you can see a blood film from a patient with leukemia, and these are the leukemic cells. They proliferate in the bone marrow, take over the normal bone marrow, and spill over into the peripheral blood. And on the right-hand panel, you can see two tubes of blood. The left-hand one is what our blood would look like if we took a blood sample and sat on the bench. Your red blood cells would fall to the bottom, the plasma would sit on top, and across here you can see a hazy layer. That's the white blood cells. Here is a blood from a patient with leukemia. Here's the red blood cells, so a lot less of them. Here's the plasma, and this layer here are all the leukemia cells. And in fact, when you look back into the historical medical literature, this is how leukemia got its name. Leukemia literally means white blood because of these leukemic cells in the blood. As I said, the leukemia is a cancer of the bone marrow and blood. It starts off in the bone marrow. Here on the left is a normal bone marrow, so that's what yours and my bone marrow would look like. Here's part of the bone. Here is the bone marrow. These are fat cells, and this is the developing blood cells. Here on the right is a bone marrow that we've taken from a patient with leukemia. So you can see in pink the little bone fragments, but here it's just replaced. All the normal developing blood cells are replaced by this infiltration of leukemia cells. As a consequence, the patients that we look after when they present have signs and symptoms because of that failure of the bone marrow. So patients are anemic, they have a low hemoglobin, so there's less oxygen carriage around the body, and so presentation is with tiredness, fatigue, shortness of breath on minor exertion. There's a failure of production of the normal white blood cells, our infection-fighting cells. So we call these neutrophils. Patients present with infection. I've shown here's a chest x-ray of a patient who presented and had pneumonia. You can see there's additional white material in the upper lobe of the left lung. And here on the left-hand panel is just an ongoing fever in a patient with leukemia. And finally, patients may present with abnormal bruising and bleeding, and that's because the platelet count, that's the clotting cells, are low. So here you can see bruises and these little fine petechiae, and often they're over the foot. That's just a gravitational thing. And here in the mouth and the mucosa, and you can see bruising and bleeding here. So this is the issue that patients with acute leukemia present with, and the problem we have when we're managing these patients. When we look at the field of personalised haematology, is that up until recently, we treated all patients the same. So the presentation is similar, clinical features are similar, and patients would be treated usually with curative intent, with the goal of putting the patient into remission and resulting with a cure. But patients would all receive the same type of chemotherapy, but there was variable outcomes. Not all patients went into remission. Some patients would go into remission and then would have a subsequent relapse. So the goal of our research is to dive down deeper and try and find out what is the difference between these patients with acute leukemia, and how can we individualize therapy for each patient so that we can improve their outcomes, potentially reduce the side effects of therapy. So the goal of this is to work around understanding the genetic changes that occur in the leukemia. And so we started, and over 2 decades ago, looking at the chromosome changes. So this is in the DNA of the cell. So this is the part of the cell that controls all the function and activities of the cell. And so we recognize that the blood cancers, and particularly the acute leukemias, often had characteristic chromosomal abnormalities. So I've shown an example here from one of our patients. Here's all the patient's chromosomes. Here's the sex chromosomes, the two X chromosomes in this young woman. And you can see there's an abnormality between chromosomes 8 and 21. There's a swapping over of material. And so this is a translocation between chromosomes 8 and 21. And about half the patients we see with acute leukemia have chromosome changes, and that does help predict for outcome, and in some cases may also alter our therapy and our approach. what I'm showing here is this is what we call a Kaplan-Meier survival curve. So this is a statistical tool that we use in research and in the clinic to compare the outcome for different patients according to either their chromosomes or what treatment they have. So on this y-axis on the left-hand side of the panel, you can see here's the percentage of patients that are remaining in remission, and here on the x-axis is the years following their diagnosis and treatment. And you can see there's quite different outcomes based on these chromosome changes, with some chromosome changes having a very good outcome, other patients doing less well with a high risk of relapse, and often will alter therapy based on these findings. The problem is that not all patients have chromosomal changes, but more importantly, the chromosomal studies will only detect changes in the number of chromosomes and the structure of chromosomes. So if we think about the nucleus of a cell, the DNA in the nucleus, so the genetics of that cell, it's a bit like having a library that's got a series of books in it, and all the words in those books have the controls and the mechanisms that oversee the development of the cell, the maturation of the cell, and what the cell does. So chromosome studies allow us to look to see how many books we've got in our library and if any of them are missing, or if one of the books is in the wrong place. But what it can't tell us is that if there are small mutations, small changes within the book, so if there is a spelling mistake on one of the pages in the book, or even if several of the pages are missing, we can't tell by just looking at the library. We need to be able to look deeper. We need to be able to look at the pages. We need to be able to look at the words. And this has been the huge advance in the cancer area and the blood cancer area, and it's the area that our research has focused on. This slide shows that we're looking at the study of the genetics of leukemia. It's a wee bit like an iceberg. So up until recently, we were looking at the chromosome changes. So those are the side genetics. So that's like looking at what you can see above the water, but we know the bulk of the changes, the bulk of the iceberg is below the water. And that's this field of molecular genetics, that ability to look at those spelling mistakes, those small mutations. And science and technology have moved to allow us to do it. So here we've got the chromosome changes. Now with the ability to sequence through genes, we can look at all of those small spelling mistakes. So around just over a decade ago, we started looking at individual genes, but the technology only allowed us to look at a small number of genes. So in the blood cancer area, it was these 3 critical genes, and that was useful, but there's over 100 genes are important in the development of leukemia. So we were looking at a very small number of the genes for potential mutations. In the last decade, we've seen huge advances in technology. So here we've got where we used to sequence the gene, and we could look at a small number of so the bases are the single coding points, so those are the small changes we're looking for. This is how many we could look at, around 600 per day. Now with modern technology and what we call next generation sequencing, with this platform here in our lab, we can look at the whole human genome in a 24-hour period. To put that into perspective of technologies that we understand better, is that's like comparing the advances from a foot-powered bicycle through to the Starship Enterprise at warp speed. And so this development of technology has given us so much more information around the genetic changes that we see in leukemia and other cancer cells. So our study, the Auckland Myeloid Gene Panel study, has been not only to look at the number of books in the library, but to look at each page for any missing pages and spelling mistakes that might occur within those books. So within the DNA, within the controlling pathways for the leukemia cells. We developed a panel, initially a 78 gene panel, so genes that are involved in blood cancers. We've now expanded that into 110 genes that we look at. So the goals of our study were to look at, were the particular mutations within the leukemia cells that might change a protein that we could then target with a specific therapy or drug. Secondly, we were looking for information that might guide treatment and might alter the prognosis, might indicate a more favourable prognosis where we could perhaps use less intense treatment, or a more unfavourable prognosis where we would get better outcomes giving more intensive and aggressive therapy early on. And thirdly, we were interested in looking at molecular biomarkers so that we could follow the patient to assess their response to treatment. This just shows the type of report that we generate from our myeloid gene panel study. So here we have the patient details. Here we have what genetic and chromosome and morphology information we have. And here, after looking at the genes, here are the mutations that we've identified within this patient's leukemia. So each patient going on to the study and having this analysis, we were generating, in addition to the conventional information around what the leukemia looks like under the microscope, what chromosome studies they have. We're looking at the results initially from 78 gene analysis and now 110 genes being analysed. So one of the first things that we found was that every patient was different. So a cancer that under the microscope looks similar and we treat it in a similar way, in fact at genetic level, are quite different. So if you look at the right-hand panel, across the top are just over 20 patients that have been analysed, and down the left-hand panel are the different leukemia genes we look at, and the yellow marker indicates a mutation within one of those genes. And what you can see is that each patient has got a different molecular profile. So therefore it's not surprising that if we treat these patients all in the same way, we're going to have different outcomes. So large international studies, and this is one that we contributed to, have shown that in addition to the chromosome changes, different molecular changes may also predict for the outcome for a patient. So this is, again, these are probability of remaining in remission curves, and these are different mutation profiles that we see. So you can see some predict for a very good outcome, whereas other patients there's a higher risk of relapse. So again, moving to this genetic analysis of the leukemia cells is providing more information. It's empowering the patient, it's empowering the clinician, it's giving more information to guide our therapy. We looked at the first 100 patients that were analysed as part of the myeloid gene panel study, and this first slide shows you the data on the number of mutations that we detect in each patient. So across the x-axis is the number of mutations seen, and on the y-axis is the number of patients that had that number of mutations. A small number didn't have any mutation, but they had characteristic chromosome changes. The mean number of mutations seen in each patient was just over 3. So these are critical mutations, so critical spelling mistakes that contribute to the development of the leukemia. This just shows the pattern of mutations that we see. So these are all the genes that we looked at. So these are genes that are mutated in the leukemias, and these are the number of patients that had those mutations. So you can see some mutations occur more frequently than others, whereas others are quite infrequently seen. So these ones here are really important and important in the development of the leukemia. So these are ones that we've focused on and have looked at in more detail. We then looked at, well, did doing this initial and now additional analysis, did it change our treatment? Did it benefit the patient? Did it change an outcome? So what we're looking at here is the classification of these leukemias into good risk, an intermediate risk, and unfavourable risk. And on the grey are the classification based on the conventional information, so clinical information, the age of the patient, their white cell count, chromosome changes. On the right, in the orange colour, are the changes or the new risk groups based on adding in the molecular classification. And you can see just under 20% of patients had a prognostic subgroup change. One patient who was being considered for more intensive treatment and potentially a stem cell transplant moved into a more favourable risk group, where we know there's a good chance of cure with chemotherapy alone. The majority of patients had molecular risk factors that moved them into a more unfavourable risk group, where we knew there weren't good outcomes with conventional chemotherapy, and we'd look at either experimental therapy or perhaps an early stem cell transplant to improve outcome. In 50% of patients, we identified a mutation that would change a protein within the leukemia cell that would be a potential target for drug therapy. What do I mean by that? So where the mutation changes a protein that's involved in a critical pathway, perhaps a pathway that makes the leukemia cell or the cancer cell grow more rapidly, then we could target that. So if we've got a drug that can target that pathway, we'd call that a druggable sort of target. Here is an example. In 10% of our leukemia patients, we find a mutation within the IDH2 gene. There is a drug, enasidenib, that targets the mutant IDH gene protein. And here's the preliminary data. This was early data from patients with relapsed acute myeloid leukemia carrying an IDH2 gene mutation. They were treated with enasidenib, and around 40% of patients had an improvement in their blood counts and their bone marrow appearances. Based on these results, there are now large international studies that are ongoing, and we're contributing patients to these, where we test the genes early on before treatment starts. If they've got an IDH2 mutation, then where the trial is comparing conventional chemotherapy with the IDH2 inhibitor or a placebo to see if there is a benefit of adding in an IDH2 inhibitor. Our final finding was that in the majority of patients, we could identify a mutation that we could use as a biomarker for monitoring therapy and looking at the response to therapy. So what I mean by that is that when we treat a patient with leukemia, is that we define remission as that there's recovery of their blood count. So remember back, we were talking around the symptoms that a patient has with leukemia, so we look for essentially a normal blood count, a normal hemoglobin, normal white blood cells, normal platelets. We do a bone marrow test and we look to see if it's in remission, and remission is defined as having less than 5% blast, so less than 5% leukemia cells in the bone marrow. We know that when patients are in remission by these conventional criteria, that there is still a significant burden of leukemia cells, and that we give ongoing therapy to keep the patient in remission. What the goal of our biomarker therapy is, that can we use molecular techniques to monitor down to a much deeper level. So one of the genes that we look at is the NPM1 gene, which if we amplify that in the laboratory, we can detect not to a level of 1 in 20, but to a level of 1 in a million. So we call this minimal residual disease monitoring or measurable residual disease monitoring. So this has moved into our clinical practice, into our ongoing clinical trials of identifying a biomarker and following that during treatment to determine the depth of remission. And the data is telling us that the lower your level of minimal residual disease, the better the outcome. And an analogy here is that it's a bit like the Where's Wally cartoon. So you're looking amongst a mass of blood cells for the one cell that might be carrying this leukemia marker. And so that's the goals of our therapy. And we have an ongoing trial with clinical colleagues across Australia and just starting in New Zealand, where we're taking patients who are in remission, we're doing the molecular monitoring, and if we see that biomarker go from negative to positive, we're asking the question is, can we start a targeted therapy early before the patient's had a full relapse, and will that benefit the patient? So the treatment's moving from the traditional morphology of what's happening in the blood and what's happening in the bone marrow to a molecular monitoring. And an unexpected finding from our research was that within the blood cancers there is a higher incidence of a familial predisposition than we initially anticipated. So you'll be familiar that in the solid cancers, that particularly in breast and colorectal cancer, that about 5% of those, there are inherited genes that predispose to the development of those cancers. The traditional teaching in the blood cancers was that inherited predisposition was relatively uncommon. The mutations that I've talked about to date are all what we call somatic or acquired mutations. So you're not born with them, they're just in the leukemia cell or the cancer cell. What we found in this study is that amongst the first 200 patients that we looked at, that we identified 8 index cases where there was an inherited or germline predisposition. One was in a gene that's been well recognized as being inherited, CBP alpha, and in this patient there was a positive family history. But we also identified many cases of a recently described gene called DDX41, where there was an inherited mutation, and the majority of these patients didn't have a family history and were an older age of onset of the leukemia or blood cancer. So this is an example of one of the cases that we identified. So this was a 52-year-old male diagnosed with acute myeloid leukemia, and here are the different mutations that we identified on the bone marrow DNA analysis, so the leukemia cell DNA analysis. And you can see that there are 2 mutations in this gene that I've just spoken about called DDX41. And there's this mutation, this is inherited. So when we tested non-blood tissue, so in this case we took eyebrows and we extracted the DNA from the eyebrows and analyzed for a DDX41 mutation and found that it was present in the DNA. And then the second mutation, this is an acquired mutation. So that was we all have 2 copies of this gene, so this one occurred within the leukemia cells. So this is an inherited mutation of DDX41. Why is this significant? So it may impact on the treatment recommendations for the patient. There's potentially other family members who may have inherited this gene, so potential for testing following genetic counselling of those family members. And in this patient, we were considering a stem cell transplant from a family member as part of the therapy. So it'd be important to ensure that the donor didn't also carry that mutation that predisposed to leukemia. This is a publication from the literature from a patient, this patient here, 2C, who had acute leukemia and carried a DDX41 mutation, and he had a transplant from his brother, and the brother also carried a DDX41 mutation. The patient had a relapse at a later time, not of his leukemia cells, but in fact it was in the donor cells. So it'd be really important if we've got a patient who's got an inherited mutation being considered for a transplant that we exclude that the donor is carrying that same inherited mutation. So what I hope I've shown you in this brief review of our research is that when we look at personalized medicine in acute myeloid leukemia, is that when a lot of people think about doing genetic analysis of leukemia and cancer cells, they're looking at a target that they can treat, that you can use a novel treatment. But what our research has shown, it gives much more information than that. So it helps to confirm the diagnosis and subtype of leukemia, and a new classification of the blood cancers was published at the end of last year, and it is very much now based around a molecular classification. Secondly, it tells us what's going to happen to the patient, so it provides information on the prognosis. As I mentioned, in half the patients we can identify a mutation that is potentially sort of druggable, where we can look at a new therapy. In most patients, it gives us a biomarker, so increasingly we're moving to molecular monitoring, so we're not testing down to a level of 5%, we're testing down to a level of 1 in a million. And then finally, it's giving us information, is there a family risk of developing leukemia? And a totally unexpected finding in our research is that around 3 to 4% of the blood cancers that we're identifying a familial predisposition. This work is in the blood cancer area, but I think the findings here also become applicable to other cancers, and there's a lot of work going on both within the University of Auckland in the precision medicine field and internationally, looking at the impact of genomic testing in cancers. So thank you very much for tuning in. Thank you very much for your attention. Can I just acknowledge I'm a very small part of all of this research. So this work was done in the Leukemia and Blood Cancer Research Unit within the Centre for Cancer Research in the University of Auckland. A lot of it led by Stefan Bolander, my co-director of the LBCRU, and Purvi, who's the scientist who's driven through all our molecular work. We also work closely with our colleagues in molecular haematology at Auckland Hospital and our clinical colleagues at Auckland City Hospital who care for these patients. And finally, the patients and their families who will consented to having their leukemia DNA analysed as part of this study.
Hello, I'm Nuala Helsby, and I'd like to talk to you today about my research interest, which is cancer pharmacogenetics. Over the years, we've understood more and more about the cancer genome and the importance of the genome in both prevention of cancer and also our improved understanding on how to treat cancer. And by the genome, I simply mean the DNA molecules within inside the nucleus of cells. And we've understood for a number of years now the important role of environmental chemicals that can cause damage to the DNA, and they're called mutagens. And if we can avoid those environmental damaging agents, DNA damaging agents, we can help prevent and minimize the chances of getting cancer. So that's an important part of our understanding of the cancer genome. Over the years, we've also understood, though, that certain cancers can run in families, and these are called inherited cancer syndromes. And our ability to understand the genetic changes in the DNA in those individuals within families help us not only understand who's going to be at risk of developing some of these cancers, but also developing screening programs to try to minimize or prevent the risk of people developing cancer. Over the years, we've also improved our treatments of cancer by understanding more about how, the cancer genome, the DNA inside cancer cells. And one of the things that we do know is that cytotoxic chemotherapy drugs and radiation therapy damage the tumour cells and kill the tumour cells by causing a lot of DNA damage inside those cells. But over the more recent years, we've also been able to start to understand that within one cancer type, there may be lots and lots of subtypes, and we call these molecular subtypes. And they are because within each type of cancer, there might be lots and lots of different changes in the DNA. So somatic mutations in the DNA in those tumour cells. And that might mean that some of those subtypes of cancers are overexpressing certain proteins and certain pathways. And that means we can then target those proteins or pathways with very selective new drugs that have more recently become available on the market. I'm interested in something slightly different though. So I am interested in the person, not the tumour, not the cancer. And I'm interested in something called cancer pharmacogenetics. And these are the inherited differences in how each individual person responds to therapy. And in the context of cancer, I'm particularly interested in cancer chemotherapy. So I'm interested in pharmacogenetics, and that is simply trying to study and understand the inherited differences, or differences in our genes between individuals that might mean that in some people a drug doesn't work very well, and in other people a drug might not be safe. So it's this real balance between the benefits and harms of a drug that might relate to your differences that you've inherited from your parents. Now, I work on both sides of this balance, but what I want to tell you about today is some work that we've been doing for many years now, trying to understand why one particular cancer drug might not work in some people. So what I want to talk to you today about is this drug, cyclophosphamide. Now, it's one of the oldest chemotherapy drugs, and it's given as an intravenous drip to patients. And it's really key for the treatment of cancers such as breast cancer, but also hematological malignancies, which are blood cancers like lymphoma and leukemia, and also in some of the very rare childhood cancers. Now, cyclophosphamide is this compound here. This is the chemical structure of cyclophosphamide. And it works by these two arms of the molecule attaching to DNA. And once it's attached to the DNA, it stops cells being able to divide. So that stops the cancer cells growing. So one of the other advantages of this ability of cyclophosphamide to bind or attach to DNA, to prevent the ability of DNA to then copy itself, to replicate, which is a key thing that's required for cells to grow and proliferate in your body. This ability to stop this process, stop DNA replication, means that cyclophosphamide is also very good at helping to treat certain autoimmune diseases. And those diseases are things like systemic lupus erythematosus and vasculitis. And that's because in autoimmune diseases, you've got a very activated immune system and lots of proliferating white blood cells that attack your normal tissues. So cyclophosphamide can dampen down and stop those proliferation of those very active cells. But in the context of cancer, cyclophosphamide has been increasingly used because of its immunosuppressive properties too. And in one context is where it's being used when patients have got hematological malignancies, some of those blood cancers, and one of the ways to treat that is to give patients a bone marrow transplantation. But if you don't dampen down your normal immune system with a little bit of a small dose of cyclophosphamide, then your normal immune system will start to attack that bone marrow transplantation. And that's called graft versus host disease. And it's one of the newer ways that cyclophosphamide is being used to help treat patients with cancer. It's also being used in another very, very new approach to treatment of cancer. And that approach is called CAR T therapy, where T cells are taken out of your body. They are trained and adapted to help them identify your tumour cells as foreign, and then they're put back into your body to then help target those, be able to attack and target your cancer cells. But to put these these altered T cells back into your body, you need to provide a little bit of space inside all of your immune system. And one way to do that is again to give a little bit of cyclophosphamide just before you reinfuse these T cells back into the body. And that's called a preconditioning, to give a niche or a tiny little space to help these T cells be able to repopulate into your body. So, why does this drug, cyclophosphamide, not work in some people? So, cyclophosphamide is actually a very complicated drug. So, cyclophosphamide itself isn't active, so it's called a prodrug. It has to be converted, it has no activity, it has to be converted in our bodies, in our livers, into a molecule called 4-hydroxycyclophosphamide. That then converts and balances itself with a compound called aldocyclophosphamide, and then there's one further step where aldocyclophosphamide gets converted into a molecule called phosphoramide mustard, and it's this phosphoramide mustard that attaches itself or alkylates DNA, and that attaching and alkylating DNA is what stops the DNA being able to replicate, so it stops the cell growth, but it also, in a cancer cell, will tell the cell to undergo cell death, to go under a process called apoptosis. Using human liver, that some individuals are very good at activating cyclophosphamide into this 4-hydroxycyclophosphamide metabolite. So some livers, very high activators of cyclophosphamide. Other livers are very poor activators of cyclophosphamide, and yet again some are somewhere in between, so intermediate activators of cyclophosphamide. So this activation of cyclophosphamide in the liver is undertaken by some enzymes, and the enzymes involved are called CYP2C19 and CYP2B6. Now, these enzymes, we've inherited differences in the activity of both of these enzymes. And this differences in the activity that we've inherited are simply due to something called single nucleotide polymorphisms, or SNPs, in the genes that code for each of those enzymes. So if you're not sure what a SNP is, it's basically the code for within each gene that encodes for a protein has a series of letters within it. And if one of those letters gets changed, for example, a change from a G to an A, then that is a single nucleotide polymorphism. And if you have one of these very small letter changes in the gene for either CYP2C19 or CYP2B6, then the activity of that enzyme is very low or non-existent. And because we have inherited 2 copies of a gene from our parents, one from our mother and one from our father, then the children of the parents can have either inherit 2 normal copies, which for CYP2C19 and CYP2B6 enzyme would say that they've got normal activity of those enzymes that can activate cyclophosphamide, or they might have inherited 2 changed copies of the gene from their parents, and that means that then they've got very poor activity for either CYP2C19 or CYP2B6 and their ability to activate cyclophosphamide in their liver. And some individuals will have inherited 1 normal copy and 1 changed copy of their genes, and they will have intermediate activity for activating cyclophosphamide by their liver. Okay, unfortunately, the pattern of these letter changes, these SNP changes in genes, is actually quite complicated. But all you really need to remember is that for CYP2C19, there are 2 common SNP changes that occur. And if you inherit any pattern of these SNP variants, and you inherit 2 of these copies that are variant genes with low activity, then you will be a poor metabolizer of drugs, such as cyclophosphamide, because you've got no enzyme and therefore no activity. and that's sometimes called null. For CYP2B6, though, it's a wee bit more complicated because it's a very, very complex gene. But all you really need to remember in the context of today is that if you've inherited a pattern of 2 variant SNP changes in your gene, which is called the star6 variant, then you will have low enzyme activity. And and if you've inherited essentially 2 copies of this star6, which has 2 SNPs in it, then you are also likely to be a poor metabolizer of cyclophosphamide, with very, very low activity of being able to activate the drug. And a number of years ago now, we were able to demonstrate using human liver material that individuals who had inherited at least 1 variant copy of their gene in either CYP2C19 or CYP2B6 were poor metabolizers of cyclophosphamide and had much, much lower activation of cyclophosphamide in their liver. So we've been able to show that if you've inherited a low function enzyme for either CYP2C19 or CYP2B6, that certainly within liver samples that you can see decreased activation of cyclophosphamide into 4-hydroxycyclophosphamide. But that's only the first step in the activation of this drug to form that agent called phosphoramide mustard that interacts with DNA and causes all the stopping cells like cancer cells proliferating and growing. So do these inherited differences in activation matter? Do they have a clinical outcome? And we decided to look at that in 2 different ways. And the first way we decided to look to see what was already known in the literature. So we had an extensive look through all the material that had been published in the literature. And we found that when we looked at the data, there were a number of studies, 13 studies, which had shown that in patients you could see differences in how much of that activated 4-hydroxycyclophosphamide was formed and circulating in their blood supply, ready to interact with tumour cells and kill tumour cells. But what we also found was that there were 17 studies which had shown that if you've inherited any of those SNPs in either CYP2B6 or CYP2C19, then the patients will have worse outcomes when they're treated with cyclophosphamide. So they'll have worse survival following treatment for cyclophosphamide, whether it was breast cancer treatment, leukemia, lymphoma treatment, or another blood cancer called multiple myeloma, and even worse treatment outcomes in one of those autoimmune diseases that cyclophosphamide is sometimes used for, a disease called lupus. There was very variable quality of the data. So some studies had only looked for changes in inherited differences in CYP2B6, and other studies had only assessed CYP2C19. So there is still a little bit of uncertainty in the literature as to the importance of the inherited differences in both of these genes. So we then wondered if there is this strong signal from the overseas literature, can we see the effect of these inherited differences in CYP2C19 and CYP2B6 in New Zealand patients? So we decided to look to see if we could see these inherited differences in how patients can activate cyclophosphamide in patients who were being treated with cyclophosphamide for breast cancer. And what we did was we took blood samples from the patients while they were receiving their cyclophosphamide and measured to see if we could detect the formation of that 4-hydroxycyclophosphamide metabolite in relation, and look to see the differences in that levels of that metabolite compared to their inherited differences in those 2 genes, CYP2C19 and CYP2B6. And that's exactly what we saw. When we measured the concentrations of 4-hydroxycyclophosphamide in their blood samples, we could see if you've inherited a low function of CYP2C19, you had lower metabolism, lower formation of that metabolite, lower activation of cyclophosphamide. And the same was also true of inheriting variant copies of the CYP2B6 gene too. That you have more copies, the variant copies you have, the lower the bioactivation of cyclophosphamide. It's well established that across different geographical regions of the world there are different prevalence of genetic variants, and it's fairly well understood that people from parts of Asia and Southeast Asia tend to have a higher prevalence of variants in the CYP2C19 and CYP2B6 gene, and that means they are more likely, people from those regions of the world are more likely to be poor metabolizers of drugs like cyclophosphamide than people of European ancestry. Now, we don't know very much at all about the prevalence of these gene variants in people of Maori and Pacific ancestry, but in our study what we did note was that people who self-identified as not Pakeha had much lower activation of cyclophosphamide than individuals who identified as Pakeha New Zealanders. Unfortunately, it's a little bit more complicated, and that's because there is some other factors affect the enzyme activity of those cytochrome P450 or CYP enzymes that are involved in the activation of cyclophosphamide. And that's because there's increasing awareness that other factors, environmental factors, can interact with your genes to decrease the activity of these enzymes. And one of those environmental factors is the body's inflammatory response to the presence of a tumour in your body. And in this case, you might have inherited normal copies of the gene, so you should be a normal metabolizer, but the environmental effect of all this inflammation that occurs in your body is it down-regulates, or essentially switches off how well that gene is processed, and so you become a poor metabolizer. And this difference between your genes, saying you're a normal metabolizer, and your body's actual process, where you're a poor metabolizer, is called a phenoconversion. And so importantly, your genotype will not predict your phenotype when you've got a large inflammatory response. And these individuals are sometimes called discordant from their genotype. Now, CYP2C19, which is one of those enzymes that's important in the activation of cyclophosphamide, is particularly vulnerable to this downregulation or changing the activity of the gene for CYP2C19. And we've been able to show over a number of different studies over the years that there are a substantial number of cancer patients who are discordant for CYP2C19. So they've got a normal CYP2C19 genes, but they've got poor metabolism with this enzyme. And we can do that by probing the activity of this enzyme with probe drugs for CYP2C19. And we've done that over a number of different studies, and typically it's about 25% of patients at any one time have very, very low CYP2C19 activity that is nothing to do with their genotype. So it is a little bit more complicated than just simply your inheritance. So CYP2B6, in contrast, just doesn't seem to have the same gene environment effect. And that means that after a patient has had multiple cycles of chemotherapy treatment with cyclophosphamide, that their CYP2C19 gene no longer predicts whether they're going to be a poor activator of cyclophosphamide or not. However, the inherited differences in the CYP2B6 gene still strongly predict which women are going to have low activation of cyclophosphamide. So I hope I've been able to explain to you and persuade you that that first initial step of converting the inactive cyclophosphamide into an active compound by the human liver does involve these two enzymes, and is particularly inherited differences in CYP2B6 are probably very important in patient outcomes. But once this 4-hydroxycyclophosphamide metabolite has been formed, it readily forms another compound called aldocyclophosphamide. And this it's complicated because this metabolite can then be detoxified. It can be removed from the body by an enzyme called aldehyde dehydrogenase. And so this enzyme also has a lot of inherited differences in its activity, and this is just not well studied in any population anywhere in the world, and is something we still need to understand more about. And it's important because we do know that tissues that have high levels of this enzyme, aldehyde dehydrogenase, are protected from the toxic effects of cyclophosphamide. And that's how some of the bone marrow stem cells are protected from very high concentrations of very high doses of cyclophosphamide. But once aldocyclophosphamide has been formed, the way that then can convert into phosphoramide mustard, interact with the DNA, stop DNA being able to copy itself, replicate, and let cells, cancer cells in particular, proliferate and grow, has always been assumed that this step is a simple chemical conversion and that it doesn't involve any enzymes. However, we've very recently been able to show that we think that this final step is actually an enzyme catalyzed step 2. So, as I said, it's always been assumed that this conversion of aldophosphamide into this final reactive compound that interacts with the DNA, binds to DNA in a process that's called alkylation, that this process has always been assumed to be a simple chemical reaction, simple conversion. And we think that we've been able to show that it's actually an enzyme reaction that occurs within cells. And we've been able to do that by, first of all, we can incubate this compound, phosphoramide mustard, or 4-hydroxycyclophosphamide, just with naked DNA. And you can see that the phosphoramide mustard has very efficient binding at low concentrations to DNA, and this measure here on the y-axis is just a measure of how well it binds to the DNA. And in contrast, 4-hydroxycyclophosphamide is just not very good at binding or alkylating DNA. However, if we put the same two compounds, 4-hydroxycyclophosphamide or phosphoramide mustard, in and incubate these two compounds with peripheral blood mononuclear cells, so the white blood cells from your blood, there is no difference in how well these two compounds can alkylate or bind to DNA. And finally, we've been able to also show that if we incubate 4-hydroxycyclophosphamide with a cell line called Jurkat cells, which are a type of cells that grow in culture that are related to T lymphocytes, that we can see conversion of 4-hydroxycyclophosphamide into something that can quite efficiently bind to DNA and alkylate DNA, whereas when we put the same concentrations of 4-hydroxycyclophosphamide with another type of cell called KCO2 cells, which are epithelial cells, that we don't see this cellular activation of 4-hydroxycyclophosphamide into this DNA reactive compound. So we think we're seeing intracellular activation of 4-hydroxycyclophosphamide, and that implies that this is a cellular enzymatic reaction. What we've also been able to do is put 4-hydroxycyclophosphamide with some purified phosphodiesterase enzymes and then measure the amount of phosphoramide mustard formed, because we can measure the amount of DNA damage, DNA alkylation that we can observe in our samples. And what we've been able to find is that phosphodiesterase enzymes such as cyclic AMP phosphodiesterase can catalyze this reaction. And so that suggests that there really is an enzymatic activation of this final step from aldophosphamide to phosphoramide mustard. Now, we don't know yet how this relates to the activation of aldophosphamide to phosphoramide mustard in tumour cells, and that's something we still need to learn more about. As I mentioned right at the beginning, there's increasing use of cyclophosphamide to help moderate or suppress the immune system. And there's a lot of suggestion in the literature that this is working through a way of regulating a certain subtype of T cells in the body called Treg cells. And we're speculating that if this final step of activation of cyclophosphamide, this conversion of aldocyclophosphamide into phosphoramide mustard, which is the key agent that alkylates and binds to DNA, if this final step is catalyzed within white blood cells, so within immune cells in the body, and if it's selectively activated by this phosphodiesterase enzyme, which occurs in high levels in a lot of white blood cells, especially in T cells, that maybe this is another way in which cyclophosphamide is having this ability to be a very good immune modulatory drug, as well as a chemotherapy drug. And so there may still be more to learn about how cyclophosphamide works in the context of the newer immunotherapy treatments for cancer. So there may still be another reason why some people respond very well and very efficiently to cyclophosphamide and other people don't. So in addition to inherited differences in how people activate cyclophosphamide by their liver, there is the possibility, that we all have differences in how efficiently we repair the damaged DNA that is produced once phosphoramide mustard alkylates and binds to and damages the DNA. And in our current studies, we can detect more than a 200-fold range of how much DNA binding DNA adduct formation with phosphoramide mustard occurs between individuals. Now, this work hasn't yet been peer-reviewed and published, so I don't want to show you those graphs at the moment, but I just do want to say that there may still be another reason, potentially inherited reasons between individuals, as to how efficiently we can repair the damage that phosphoramide mustard causes. And so some people might be a bit more resistant to cyclophosphamide than other people. So I hope I've been able to persuade you that there are inherited differences in how cyclophosphamide is activated between individuals, and that understanding those inherited differences might help us understand why some people benefit from this drug and that others don't. But despite this drug being used since the 1950s, so it's really long established use, I think there remains a really important opportunity for us to start to think about maybe personalising or precision use of this medicine in certain people, both and importantly as a classical chemotherapy, cytotoxic chemotherapy agent, but also in its use in the more modern context, in its ability to modulate some of the newer immunotherapies that are being used. Thank you for you listening to me, first of all, but also thank you, a very big thank you to the numerous funders of this research over many years, including the Auckland Medical Research Foundation. I also need to say a big thank you to all the patients and tissue donors over the years, and obviously also to my numerous colleagues and research students that have been a key part of all of this research.